Hello and welcome to this webinar on best practices for validating chemical probes. This was created by the Best Practices in Medicinal Chemistry Working Group for the European Federation for Medicinal Chemistry. And as you see on this slide, this is a group of people from different pharmaceutical companies as well as academic institutions. So what we'd like to do today is go over a set of very simple guidelines that can help you validate chemical probes so that it can be useful to study human biology and disease. So let's start from the beginning. What is a chemical probe? So a chemical probe is a selective small molecule modulator of the function of a biomolecule. Usually it's a protein, but it can also be RNA, DNA, or any other biomolecule. And these will be used to study the biology associated with this biomolecule. It will allow you to ask mechanistic and phenotypic questions about this molecular target in a variety of settings such as biochemical, cell-based, or animal studies. We consider that they are complementary to genetic approaches such as siRNA or CRISPR, which are also commonly used to study the function of biomolecules. And it's important to distinguish a chemical probe from a drug. A chemical probe is designed to selectively address a biological question, whereas a drug is designed to produce a desired clinical outcome. So of course the properties that we need to have a good chemical probe will be different from that of a drug. And this is what we would like to talk to you about today. So why do we want chemical probes? Well, they can have a major impact. They are very often used to further our understanding of human biology and disease. First, they can be useful to annotate functionally the human genome to understand what the role of a protein associated with a gene can be, and understand physiological and pathological processes associated with a particular protein or gene. They can also be useful to enable drug discovery. Very often, these chemical probes can be used to validate new drug targets by making the link between these targets and a disease. They can also help set up assays, as well as validate drug discovery flowcharts. And finally, they can be used as starting points from drug discovery. One can optimize these chemical probes towards compounds that can become drug candidates. And there are many examples of very useful chemical probes. We've selected a couple of examples taken from this review cited on this slide. These are probes from the liver X receptor, as well as the BET family bromodomain. What made these very good chemical probes was, first, that they were well-characterized compounds, and also, importantly, that they were made openly available to the scientific community. The scientific community could then use these to generate insights into the role of these targets, LXR and BET bromodomain family, and understand the role they could play in disease. Ultimately, in both of these cases, this led to industry selecting these targets as potential drug discovery targets. And in both cases, you have now compounds in advanced clinical trial for the treatment of a variety of disease, which hopefully will be useful to treat a number of patients. Now, the reason we want to go over guidelines today is that there are problems associated with chemical probes. The literature is actually polluted with poor quality chemical probes. This can be because they have low or unknown selectivity. Sometimes these compounds are claimed to be selective, but they're not selective. And you can easily understand that if using a chemical probe to study a particular target, but it's touching many other targets, if it's not selective, then the biology you would uncover can be put into question. Other probes can be poorly characterized for the utility to study, for example, cellular processes or in vivo effects. They can even be promiscuous compounds which are either reactive or interfering with assays. Very often, chemical probes can have poor solubility, which can cause unspecific tox effects, and we'll come back to that later in the webinar. Again, such properties can put into question the results that you get from using these chemical probes. And most unfortunately, sometimes the probes have an uncomfort mode of action and actually are not even engaging the target they are claimed to engage. And this has led to terrible misusage of chemical probes in the past, and this is continuing today. You can imagine that once a probe is published, various scientific groups will start using it. And even if somebody publishes deficiencies of a probe, it's not a given that the probe will stop being used. In order to do that, you have to assume that the people using the probe will have read this particular paper, which is often not the case. So there are still really suboptimal probes that are used even after these deficiencies are published. And I think everybody who has been working for a while in industry or academia has once spent a lot of time and resources devalidating a particular probe just to realize that it was not useful. So it's essential that as a community, as a scientific community, we ensure high quality of our chemical probes. These are a few examples of probes which had led to problems. We talked about unselected compounds. Staurosporin on the left is one of these examples. It was reported as a PKC inhibitor, and it is a PKC inhibitor, but it's not selective. There are more than 8,000 publications using this compound, some of which are using it as a selective PKC inhibitor. And you can imagine that the results you get out of these studies cannot be used very well. The compound in the middle, iniparib, is an infamous example of a compound that went very far. It was developed as a PARP inhibitor up to clinical trials phase 3, so very far in clinical development. There's more than 1,000 publications using this compound. However, it was found later that this compound actually non-specifically modifies cysteine-containing proteins, and it's not even clear that it's actually inhibiting PARP. As a chemist, looking at the structure should raise a flag. Can such a structure actually be a specific inhibitor? The compounds on the right, gossypol and apogosypol, are other examples of compounds not engaging their targets. They were used as BH3 BCL2 PPI inhibitors in many papers, but it's now likely that they're not even engaging BCL2, but acting via other mechanisms. Again, in the eye of a chemist, this structure should raise a flag. So this is where all of us can help. As chemical biologists, as medicinal chemists, we can make the link between synthetic chemists who make the compounds, but may not be aware of biological details. In some cases, we are even the one making these compounds. And we can make the link to the biologists and physicians who know very well how to use compounds, but may not consider critical chemical issues. So it's essential that we all work together to make sure that the compounds we use to study biology and disease are appropriate. Again, as a chemist, please use your experience. Please use the chemist eye to see whether the structure raises a flag. This is another example of a compound called terfostin 23, which was reported as an EGFR inhibitor quite a while back. It is still actually sold by several vendors, including Sigma Aldrich, and there's more than 1,200 citations, including recent ones, which use this compound as a kinase inhibitor. Now, when you look at this structure, you should ask yourself, can this be real? And it's actually very close to the structure of one of the ingredients of tear gas. And you should ask yourself, well, can this actually be a specific inhibitor? So we come now to the guidelines. And one important point to make at this stage is that every guideline we're going to talk about today is just a guideline. It's not a restrictive rule. Validating a chemical probe is not a tick box exercise where you need to fulfill every single guideline we're going to talk about today. You should think about what you want to use your probe for, and then make sure the data you have generated supports this purpose. And we'll come back to that at several stages during this webinar. So the guidelines can be separated into 4 big buckets: potency, target engagement, selectivity, chemistry, and context. This was actually taken off one of the very important reviews in the field, which is also cited on this slide. So let's start with potency and target engagement. So obviously, when you talk about potency on a particular target, it should be as high as possible. But the guideline depends on the context and the target. We believe that the key is actually to demonstrate target engagement, which means in the context of the assay in which you test your compound, is the compound sufficiently engaging the target? It's not sufficient to see a functional effect in an assay. We need to make sure that the compound binds to its target. Binding and function should go hand in hand. And this is whether you talk about a biochemical, a cellular, or an in vivo experiment. So let's go a little bit into why that is important. The effect of a compound in an assay is actually not always linked to target engagement. An effect in an assay could be due to many other things that are intrinsic to your sample or to the compound structure. Just to give you a few examples, if you have impurities in your compound, in your sample, such as strong acids like trifluoroacetic acid after purification, or heavy metals because you used a catalyst in one of your reactions, this can interfere with the assay readout. You can also have chemical features in your compound that would lead to assay artifacts, such as fluorescence, chemical stability, or reactivity. All of these can actually interfere and lead to data that is not linked to specific binding of your compound to its target. And finally, one important point to always check is whether your compound leads to unfolding or aggregation of the biomolecule. This can be due to intrinsic compound properties which have nothing to do with specific binding. And this is illustrated on the right. Here you have a compound that formed these blue macromolecular aggregates. These aggregates can actually sequester proteins such as enzymes. If that happens, in your assay you will see an apparent inhibition of this enzyme. But it's not because your compound has actually specifically inhibited this enzyme, but just because it forms aggregates that sequester it. So you need to make sure you have actually target engagement and specific binding in order to be able to trust the functional effects of a compound. So let's go into how you can do that. First, starting with the most simple assays, the biochemical assays, and how to use biophysical validation. So for biophysical assays, potency is suggested to be below 100 nanomolar. Again, this is just a guideline. It depends on the particular target. And this is usually in a functional assay looking, for example, at fluorescence or other readouts. Always important is to see concentration-dependent effects. And as we'll mention for many other applications, it's always useful to use structurally related negative controls, as well as demonstrate SAR, structure-activity relationship, in order to make sure that the activity you see is specific to your particular compound. Once you see a functional effect, you need to complement it with a target engagement readout. And the good news is that for biochemical settings, there's really a plethora of biophysical techniques that have been developed to do that, some of which are listed here. Just to show one very simple example that is accessible to many labs, this is what we have put at the bottom of this slide. It's a technique called differential scanning fluorimetry. In this experiment, we use the simple concept that upon increasing temperature, protein will unfold and then aggregate. And you can actually follow this process by introducing a dye, such as Cipro orange. This dye does not fluoresce in the presence of water, but as you heat your protein and it starts to unfold, the dye will bind into the hydrophobic pockets created by the unfolding. And so you can monitor an increase in fluorescence to follow this unfolding. If you continue to heat your sample, the protein will aggregate and the dye will dissociate and you will lose this fluorescence. And you can estimate a so-called melting temperature as the 50% increase of fluorescence. And this is very useful because binding of a compound to a protein will change its stability and so its melting temperature. So you can very easily monitor the binding of a compound to a protein. As mentioned, this is just one technique. There are many other ones which are summarized in this biophysical toolbox, and we of course encourage you to have a look more specifically at each of these techniques. Okay, let's go now to cellular assays, which are a little bit more complicated. So potency cutoff are usually a little bit higher, around 1 micromolar. Again, it depends on the target. This is just a guideline. This is usually a functional readout using imaging or a pathway engagement. And again, we want to see concentration-dependent effects, and if possible, use structure-related negative controls and demonstrate ACR. Now, target engagement is sometimes more difficult, but it's still critical. And over the past few years, many groups have worked on developing techniques that actually can be very useful to measure target engagement in cells, and there were some good reviews written on the topic which are cited on this slide. Again, we'd like to present one example, which is called CETSA. It uses a similar concept as the one before, the SF. It's a thermal shift assay that uses the properties of proteins upon heating. But this time we can do this using whole cells. So in this experiment, what we will do is we will heat the cellular samples, and we will separate proteins that are still folded to ones that have been aggregated by simple centrifugation. Now, if you add a compound, such as the cartoon in the middle, you will stabilize the target protein, which is represented by this blue Pac-Man. So this will lead to a higher amount of protein that is still folded. And when you separate the soluble fraction by centrifugation, you can actually monitor the levels of protein by simple techniques like Western blot. This was what you see then on the right side of this slide. When you compare vehicle to compound-treated samples, you should see increased protein levels at higher temperature, indicating that the compound has engaged its target and stabilized it. So again, this is a relatively simple technique that can be used in cells to measure target engagement. Let's go now to in vivo. And it's important to mention that not every chemical probe is suitable for in vivo use. There are some perfectly good chemical probes which are not. But if you want to use a chemical probe in vivo, it's important, again, that you think about a couple of guidelines. The first one is to measure blood or plasma exposure, or if needed, exposure in a particular organ that you're targeting, in order to be able to interpret your data. It's quite easy to understand that if you're seeing an efficacy at a particular time point, this should be related, for the most cases, to significant concentration of your compound. And you should have a relationship between the concentration of the compound in the organ and the efficacy. The good news is that there is no additional animals needed to measure blood or plasma exposure, and we've added a simple protocol in the appendix of this slide deck in order to do that. Another important thing to think about is to check for species cross-reactivity. Usually, we validate or we test our compounds against human targets, obviously, because we want to study human biology and disease. But it's very difficult to test compounds in vivo in humans. We usually use rodents. And it can happen that the potency of your compound varies from human to rodents simply because the sequence of the protein, for example, can be different. So you need to cross-check if your compounds are still potent in rodents. Finally, as for the other types of experiments, it's also important to think about target engagement readouts. Now, this is, again, a little bit more complicated, but there are still ways to do this. The most straightforward way is to use pharmacodynamic markers and to link them to the efficacy. So a pharmacodynamic marker is a proximal marker of engagement of your target. And a common example is, for example, for kinases. Kinases are phosphorylating substrates, and it's quite straightforward in many cases to measure the levels of phosphorylated substrates, be it in blood or in other organs. What you expect to see if your compound engages in its target is a dose-dependent effect on reducing the percentage of phosphorylated substrates. Ideally, what you want to be able to do is to link higher efficacy to an increased target engagement, in other words, a decreased amount of phosphorylated substrates. If you can do this, this is a very strong sign that the compound was actually engaging its target. Okay, let's move on now to the second bucket of guidelines around selectivity. So the whole point around measuring selectivity is to make sure that the effect you're seeing in your assay is linked to the one target that you're studying and not to another target. The other thing to think about is to look broad. First, within the target family, if you're studying a GPCR, test your compound on other GPCRs. If you're studying a kinase, test it on other kinases. There's no clear cutoff on how selective a compound could be, but there are some recommendations. For example, for kinases, which are very well studied, it's thought that a 30-fold biochemical selectivity is needed, and there's a recommendation to test at least 50 carefully chosen kinases, although there are much larger commercial panels available. It's also recommended to test outside the target family, and again, you can use the many commercial panels that are available. And finally, use chemoinformatics to assess other activities that could be associated with the chemical class. If with a simple chemoinformatics search you find out that the scaffold you're using has been before reported to act on a particular target, it may be good to test it against this particular target. The second thing to think about is the relevance of selectivity in the context in which you're testing the compound. Selectivity should always be taken, for example, in reference to the concentration tested. This is illustrated with this graph on the bottom. Here you have IC50 curves, so potency curves, for a compound on target, that's in blue, and off target, that's in orange. Here what you see is a 30-fold difference in IC50 between on and off target. That's a pretty reasonable selectivity window. However, if you test your compound at 1 micromolar, you can see that despite this high selectivity, the off-target effect may influence the readout. The other thing to think about is to test off-targets that you know may lead to the same phenotype in your assay. So for example, if you're studying immune responses and you know a particular kinase has been shown to impact a particular pathway, then test your compound on this pathway. One thing to also consider is that biochemical selectivity doesn't always translate in cells, so it can be an advantage to confirm any biochemical selectivity in cells, in particular for kinases. This is a recent example where the authors of this publication were looking for a probe for cyclin G-associated kinase, also called GAC. They found a very selective compound shown at the bottom, SGC-GAC1, in a kinome scan. A kinome scan is an experiment where you test against a large set of kinases. In fact, they had a KD of 2 nanomolar on GAC, on their target, and the closest affinity was only 110 nanomolar on RIPK2, so a 50-fold selectivity in this biochemical setting. However, they looked in cells using a nanobread technology, and they found that the activity on GAC was 120 nanomolar, but the activity on RIPK2 was 360 nanomolar. So the 50-fold selectivity turned into a 3-fold selectivity, which essentially is no selectivity at all. So this shows you that you can lose actually perceived selectivity from going to biochemical to cells. Now what the authors did is they complemented this probe with a RIPK2 active, GAC inactive compound in cells. So their probe is still useful because it's complemented with another compound that can help distinguish the biology of GAC and the biology of RIPK2. Another couple of words on selectivity. It's always useful to control the effects of your probe on the target. For example, some kinase inhibitors are known to lead to decreased protein levels. So they not only inhibit the enzyme, but also decrease protein levels. And it's important to know this if you want to specifically learn the role of the enzymatic activity. As we mentioned for potency, it's important to use inactive controls. The ideal inactive control is the inactive enantiomer because it has the same 2D structure. And if you have one active enantiomer and an inactive enantiomer, this is a very good sign that the effects you see are specific to your target. And this is a famous example of the BRD inhibitor plus JQ1 and its inactive enantiomer, which are always used in pair to study gene expression. Gene expression, of course, is a sensitive readout, and by using the two compounds hand in hand, we can really distinguish what's due to BRD inhibition and what's due to the intrinsic chemical structure. Okay, let's move to the third bucket, chemistry. So chemistry is everything that has to do with chemical structures and properties. We mentioned this in the introduction. Looking at the chemical structure is important. And we can go a little bit further than this to say that some structural features should always lead to caution and careful data interpretation. To talk about two kinds of structures, the first one is called PAINs, or pan-assay interference compounds. These are compounds that contain certain motifs which have been associated in the past to assay artifacts. This can be due to potential for redox activities, metal chelation properties, fluorescence interference, aggregation, etc. And you have some very useful sites, websites, that are shown at the bottom left of this slide, which you can use to find out whether your compound has one of these PAINs motifs. Now, what's important to mention is that if you have a PAINs motif in your structure, there is a chance that it's a false positive. But some of them are real. Some of them are true specific compounds binding to their target. So you shouldn't discard a compound just because it has a PAINs motif, but it should make you more cautious and maybe do additional experiments to validate your probe. The second kind of groups that should be looked at are reactive groups. Again, because they can lead to instability, but also because they can induce stress pathways in cells. But again, it's not because you have a reactive group that the probe is necessarily bad. Actually, some reactive covalent compounds are extremely useful if they are linked to a specific mode of action, and in particular, for otherwise challenging drug targets. There's actually many excellent covalent reactive compounds that are probes and even drugs currently in use in humans. A famous example is shown on the right, ibrutinib, which is an approved covalent Burton tyrosine kinase inhibitor and blockbuster drug. And you see here this acrylamide, which covalently labels BTK, but does this in a rather selective way. Now, obviously, proof of chemical structure and purity is critical. And you can do this using simple techniques like NMR or MS. This may sound trivial to you chemists, but many commercial probes actually are poorly characterized mixtures. So it's important we ensure this is done properly. We should look at stability in relevant media, for example, assay buffers or formulations for in vivo use. This is in particular true if you think that your structure can be unstable. And as mentioned before, it's important to measure aqueous solubility because low solubility can make data more difficult to interpret, in particular potency and selectivity. You can understand this by thinking that if your solubility is lower than the potency you measure, you cannot be sure that there's enough compound to elicit the effect. Now, as we said for several other guidelines, it's not because a compound has a low solubility that this necessarily is a bad probe, but you should at least check it in order to make sure you have proper validation. So these are the most important points: purity, stability, and solubility. You can look at other interesting parameters, such as permeability. If your target is intracellular, it can be helpful to measure permeability, but it's not absolutely necessary, in particular if you have demonstrated target engagement in cells. If you want to do in vivo studies, it may be useful to measure in vitro metabolism in order to prepare for these studies. And we've added a protocol in the appendix of this slide deck to help you do that. Okay, we move now on the last part of the guidelines, which are context. And it's always important to think that all the guidelines should be always adapted to the context in which you're testing the compound. We talked about some of this. The active concentration in the assay should be considered in relation to the selectivity of your compound. And if you're working on a target class that regulates genes, then look at other genes to make sure that you're not missing any important information. Once a probe is published, it's important to repeat profiling in multiple biological systems and, if possible, different labs so that you can actually strengthen your probe and refine in which context it is best used. For each system, of course, aim to demonstrate target engagement using relevant biomarkers. And this really speaks towards making your probe available to the scientific community. If you do that, you can really hope and think that other people will start using it and help you refine these properties and in which context it is most useful. So provide information on how to access it, what's the characteristics of the probe, data, and how to use it. If possible, register it on a probe portal, such as the chemical probe portal, so that other people have access to it. Several companies have actually started to have innovation portals where they make their chemical probes available to the scientific community because they think that by doing so, they will contribute to understanding of human disease and finding additional interesting drug targets. This is not the main focus of this webinar, but we'd like to say a few words about genetic methods because we think these are complementary to using chemical probes. This is depicted on the cartoon at the bottom of this slide. The chemical probe usually will perturb the function of a protein, for example, by inhibition. A complementary approach is to use genetic knockdowns by siRNA or CRISPR that will reduce protein levels. Now, if the two types of experiments have the same effect in the functional readout, this can be a very strong piece of evidence that this is linked to the function of the protein you're studying. Now, of course, not everybody can do genetic validation, and again, this speaks towards distributing your probe, because if you do, other people may do that for you. We've added additional guidelines and details on genetic methods in the appendix of this slide deck. This paper, cited here in Cancer Cell, assembled this very nice do's and don'ts for chemical probes. We won't go into all of that. These are very simple guidelines, such as consulting experienced medicinal chemists to look at the structure, rather than choosing a probe simply based on what others have used in the past. Go over it. This is a very useful exercise to do before using a probe. And before we finish, we'd like to highlight an important topic, which is the concept of preprobes. It's easy to understand that high-quality probes rarely emerge from screening efforts, and they need to be optimized. And not everybody has the resources to actually optimize these compounds further. And still, it is actually useful to publish and to share these early compounds, even if they do not fulfill the criteria for good probes, because they can be useful to others. Others can work on them further and optimize them. The important point is to make the limitation of these compounds clearly known. These are what we call preprobes. So coming back to an example we mentioned at the beginning, staurosporin, which is a rather unselective PKC inhibitor, itself, it's not a very good chemical probe. However, it was used by other groups to create more selective PKC inhibitor, such as ruboxystorin and later sotrasporin. So people were able to develop very useful chemical probes from this early preprobe. So don't hesitate to publish early compounds, as long as you make the limitations clearly known. This slide collects useful resources. The 5 first bullet points show useful web portals containing information as well as chemical probes. And we've also summarized some key literature most of which were cited during this webinar on a few slides before. Just to show two examples, this is the chemical probes portal. This is a website where you can find information on quite a number of chemical probes, and you can actually deposit your own chemical probe on these portals, and it will then be evaluated by experts and rated. This is a very easy way and good way to make it available to the scientific community. As mentioned, some companies such as Boehringer Ingelheim have set up open innovation portals. OpenMe.com is a collection of very well characterized chemical probes which you can order for free and use them in your biology experiments. So let's finish by a few take-home messages. First, don't take a published commercial probe as granted. Ask yourself, is the probe following the guidelines we've talked about today? Is it suited to answer your questions? Think about reproducing a basic set of data to build your own confidence that this is a useful chemical probe. And ask an expert before using this probe. As we've seen in several cases in this webinar, the eye of the chemist remains important to make sure there's nothing obvious in the chemical structure that should raise a flag. And use the web resources we've shown on chemical probes. Overall, the quality of probes still needs to increase, and we believe this can be achieved following the simple guidelines we've talked about today. Again, as mentioned at the beginning, these are guidelines to help, not rules. It's perfectly understandable that not everybody can generate all the data that it would be needed to fulfill all the guidelines we've shown today. First of all, it's not needed, but also it's not always possible. So adapt these guidelines to what you can do and what you think you should do to make your probe good and good enough to be published and shared with others. And if you can't generate all the data, make the limitations clear and others may generate the data you need. Finally, we've created a couple of case studies which are available on the EFMC website, and that can be used to illustrate with real stories how these guidelines can be used. And finally, please spread these messages so that the overall quality of probes and data increases. Thank you very much.